What is the real purpose of junk DNA? It's a good question. For a long time, people in science called a lot of our DNA junk. It seemed like it didn't do anything important, it didn't code for proteins. Proteins are the building blocks of life. And they do most of the work in our cells. So if a piece of DNA didn't make a protein, it was often thought of as useless. Like extra pages in a book that had no writing on them, or static on a radio channel. It was just there. It was just clutter. But that idea has really changed. Our understanding has grown a lot. Now, many scientists don't like the word junk at all. They see this non coding DNA very differently. They see it as a huge, complex part of our genetic instruction manual. It's not just filler. It's a big part of what makes us who we are, and it might be even more important than we first thought. Let's talk about where the junk idea came from. When scientists first started looking at DNA, they were amazed. They found the double helix. They figured out how DNA codes for proteins. It was a groundbreaking discovery. They saw that specific stretches of DNA called genes contained the instructions. These instructions were for making proteins. These proteins build our bodies. They make our enzymes. They help our cells communicate. It was very clear that these protein coding genes were vital. But then they looked closer. They found that these protein coding genes made up a small part of our total DNA. A very small part. In humans, it's only about 1 to 2%. That's not much. The vast majority of our DNA, over 98%, did not seem to code for proteins. It didn't have the clear, simple instructions for making a building block, so it was natural to wonder what all that other stuff was doing. It was easy to label it as junk. It was like finding a giant library. You see a few books that have clear stories, but most of the shelves are filled with random letters, or repeated words, or just blank pages. You might think those are mistakes, or just leftover bits, and some of it really might be like that. Our genome, all our DNA, has been evolving for billions of years. It's been changing. It's been accumulating things. It's been keeping things. Sometimes things stick around even if they don't have a clear purpose anymore. Like old files on a computer hard drive that you never use, but even those old files can sometimes be useful, or they can contain hidden information. So the idea of junk DNA was born from this early view. It was a practical label. It helped scientists focus on the parts they understood. It helped them focus on the parts that clearly made proteins. But as technology improved, as we could read DNA better, as we could study its effects more deeply, the picture started to change. We started to find things. We started to find activity. We started to find patterns in those seemingly blank pages, in those random letters. One of the first big clues that something was up came from non-coding RNA. See, DNA doesn't directly make proteins. It first makes an RNA copy. This RNA copy then goes on to make the protein. This is called messenger RNA, or mRNA, but we knew for a long time that not all RNA makes proteins. There are other types of RNA that are very important, and they don't code for anything. Take transfer RNA, or tRNA. These are small RNA molecules. They are crucial for making proteins. They act like little delivery trucks. They bring the right amino acids to the ribosome. The ribosome is the cell's protein-making factory. Without tRNA, no proteins could be made. So, the DNA that codes for tRNA is not junk. It's essential, even though it doesn't make a protein itself. It makes a piece of RNA that helps make proteins. Then there's ribosomal RNA, or rRNA. This RNA is a main part of the ribosome itself. The ribosome is made of both protein and rRNA. The rRNA helps the ribosome do its job. It helps it read the mRNA instructions. It helps it link amino acids together. Again, it's not a protein, but it's absolutely vital. Without RNA, no ribosomes. Without ribosomes, no protein making. So, the DNA that makes rRNA is definitely not junk. It's a core component. These were the first cracks in the junk DNA idea. These were non-protein coding parts of DNA. But they were clearly important. They were doing big jobs. But these were well understood. They were like the obvious punctuation marks in the book. You might not read them as words, but you know they are important for understanding. Then came the bigger surprises. Scientists started to find many, many more types of non-coding RNA. And these were not just structural, they were regulatory, they were controlling things. They were like tiny conductors of an orchestra. One of the most famous examples is microRNA. People call them miRNA for short. These are tiny, tiny pieces of RNA. They are really small, but they have a huge job. They act like little dimmers for genes. Imagine you have a light switch. You can turn the light on or off. But a dimmer lets you make the light brighter or dimmer. microRNAs do something similar for genes. They can make a gene's message weaker. They can stop a gene from making too much protein. They do this by binding to messenger RNA. That's the mRNA. The mRNA carries the instructions from the DNA. It tells the cell how to build a protein. When an mRNA binds to the mRNA, it can mess up those instructions. It can make the mRNA get broken down. Or it can just stop the protein from being made. This is a very precise way to control things. It means the cell can fine-tune its activity. It can react to different situations. It's like having thousands of tiny volume controls. They are all working at once. They make sure the right amount of everything is produced. Not too much, not too little. These mRNAs are involved in lots of things. They help cells grow, they help them develop. They're part of how our bodies fight off disease. When they don't work right, it can lead to problems. It can lead to sickness, even cancer. So these tiny pieces of junk DNA are actually very important regulators. They are essential for keeping our cells healthy. They're essential for keeping our bodies working. And there are hundreds of different kinds of them. Each one might target many different genes. It's a very complex network. And there are other small non-coding RNAs too, things like small interfering RNAs or siRNAs. These are a bit like miRNAs. They also help control gene expression. They often target viral DNA. Or they target jumping genes. More on jumping genes later. They act like a defense system. They shut down unwanted genetic invaders. They keep the genome stable. They keep it safe. Then there are small nucleolar RNAs, or SNORNAs. These help process and modify other RNAs. Like rRNA and tRNA, they are like quality control agents. They make sure the other RNAs are built correctly. And small nuclear RNAs, or snRNAs, these are involved in splicing. Splicing is a key step in making mRNA. When DNA makes an RNA copy, it often has bits that need to be cut out. These are called introns. The useful bits are called exons. NRNAs help make sure those introns are cut out cleanly. And the exons are stitched together perfectly. If splicing goes wrong, the protein made from that mRNA will be faulty. So, snRNAs are vital for making good proteins. So already we have a whole bunch of non-coding RNAs. And they are all doing very important jobs. They are not just sitting there. They are actively involved in the cell's daily life. They are crucial for its function, but that's not all. There are also long non-coding RNAs. People call them LNCRNAs. These are longer than the small RNAs, much longer, and we are still figuring out what many of them do. They are a big mystery, but we know they are important. Many of them are made from regions of DNA that were once firmly labeled junk. LNCRNAs seem to do a wide variety of things. They can act as scaffolds, they can bring different proteins together, they can organize things inside the cell nucleus, they can act as guides, they can direct proteins to specific places on the DNA. This helps control which genes get turned on or off. They can act as decoys. They can soak up other regulatory molecules. This stops those molecules from doing their usual job. It's a way to fine-tune activity. Some LNCRNAs are involved in epigenetics. This is a fancy word. It means changes in gene activity that don't involve changes in the DNA sequence itself. It's like adding notes in the margin of the book or highlighting certain paragraphs. The words don't change, but how you read them might. LNCRNAs can help modify the structure of chromatin. Chromatin is how DNA is packaged inside the cell. It's like winding a long string around spools. If the DNA is wound up tight, genes are hard to read. If it's loose, genes are easy to read. LNCRNAs can help determine how tightly DNA is wound. This affects which genes are active and which are silent, and many LNCRNAs are expressed in specific ways. They are found in certain tissues, or at certain stages of development. This suggests they play specialized roles. They might be important for the brain to develop properly, or for heart cells to function. When they go wrong, they are linked to diseases. Things like cancer, things like neurodegenerative disorders. So, these long stretches of non-coding DNA making these complex LNCRNAs are far from junk. They are part of the very sophisticated control system of our cells. Beyond non-coding RNAs, there are other huge categories of junk DNA that are now seen as essential. These are the regulatory elements. These are specific DNA sequences. They don't make proteins. They don't make RNA that then makes proteins, but they control when and where proteins are made. They are like the light switches and dimmers for the entire genome. Think about promoters. A promoter is a sequence of DNA just before a gene. It's like the start button for that gene. RNA polymerase, the enzyme that reads DNA to make RNA, needs to bind to the promoter. It's like the engine of a train needing to be on the tracks at the station. If there's no promoter or a faulty one, the gene won't be read. The protein won't be made. Promoters are absolutely essential. They tell the cell, start reading here. And then there are enhancers. These are incredibly important. Enhancers are DNA sequences that can be very far away from a gene. They can be thousands, even millions of bases away, but they can loop around. They can physically connect with the gene's promoter. When they do, they boost gene activity. They make the gene get read much more strongly. They are like turbochargers for genes. A single gene might have many enhancers, and each enhancer might control the gene in a different tissue, or in response to a different signal. So, an enhancer might make a gene active only in the liver, or only when the cell is stressed. This is why different cells in our body can have the same DNA, but they look and act so differently. A brain cell, a skin cell, a muscle cell, they all have the same genetic blueprint, but they use different parts of it. They turn on different genes, they turn off others. This precise control is largely thanks to enhancers and other regulatory elements. So these distant pieces of DNA not making proteins are orchestrating the symphony of gene expression. They are telling each cell what to be and what to do. There are also silencers. As the name suggests, they do the opposite of enhancers. They turn genes off, or they dampen their activity. They are like the stop button, or the mute button. And insulators. These are DNA sequences that act like barriers. They prevent enhancers from accidentally turning on the wrong gene, or turning off the wrong gene. They create boundaries. They keep things organized. They make sure the right signals go to the right genes. They prevent genetic crosstalk, so all these promoters, enhancers, silencers, and insulators are non-coding DNA. They don't provide instructions for proteins, but they are the master control panel. They decide the fate of every protein coding gene. Without them, our cells would be chaos. They would not know what to do. They would not know when to do it. Another big part of the junk DNA category is repetitive DNA. This is DNA that repeats itself, sometimes in short stretches, sometimes in very long stretches. For a long time, this was definitely seen as useless, just copies of the same thing. Over and over, why would we need that? But we found functions for some repetitive DNA. Take transposons, for example. These are often called jumping genes. They are segments of DNA that can copy themselves, and then insert those copies into new places in the genome. Or they can cut themselves out and move to a new spot. They make up a huge chunk of our DNA. In humans, they are over 45% of our genome. Imagine that! Almost half of our DNA is made of these jumping bits. For a long time, transposons were seen as parasites. Genetic hitchhikers. They just replicate themselves. They don't do anything for the host. And sometimes, they can cause problems. If they jump into the middle of a vital gene, they can break it. They can cause disease. But now, we see them differently. They are not just parasites. They are also a raw material for evolution. When they jump around, they can create new regulatory sequences. They can change how genes are expressed. They can even carry parts of other genes with them. This can lead to new genes, or new functions for existing genes. They can shuffle the deck of cards. They can create new combinations. Sometimes, a transposon might jump near an existing gene, and it might bring its own enhancer or promoter with it. This can give the gene a new way to be regulated. And it can make the gene active in a new tissue, or at a new time. This could be beneficial. It could help an organism adapt to new environments. So these jumping genes, while sometimes problematic, are also a powerful force for change and innovation in the genome. They are like a creative disruptor. Then there's satellite DNA. These are very highly repeated sequences. They often gather in specific places, for example at the centromeres of chromosomes. Centromeres are the constriction points on chromosomes. They are crucial for cell division. When a cell divides, chromosomes need to be pulled apart equally. The centromere is where the pulling machinery attaches. Without proper centromeres, chromosomes can't be segregated correctly. This leads to big problems for the cell and for the organism. So the repetitive DNA at centromeres is not junk. It's a structural necessity. It's like the anchor point and telomeres. These are at the very ends of chromosomes. They are also made of repetitive DNA. They are like caps. They protect the ends of chromosomes from damage. Imagine the plastic tips on shoelaces. They stop the shoelace from fraying. Telomeres do the same thing for our chromosomes. Every time a cell divides, a little bit of the telomere gets worn away. This protects the actual genes from being lost. When telomeres get too short, the cell can no longer divide. This is linked to aging. and to cancer. So these repetitive sequences at the ends are vital. They are guardians of our genetic information. Then there are pseudogenes. These are DNA sequences that look like real genes. They have similar sequences, but they are often broken. They have mutations that stop them from making a functional protein. For a long time, these were textbook examples of junk. They were seen as evolutionary relics, like a broken tool left in a shed. But even pseudogenes are now getting a second look. Some pseudogenes might not make proteins, but they might be transcribed into RNA. And the RNA might have a regulatory role. It might act like a sponge. It might soak up microRNAs. This could free up the real functional gene to make more protein, or it could do something else entirely. We are still figuring it out. And sometimes pseudogenes can even be reactivated, or they can give rise to entirely new functional genes over evolutionary time. So even the broken tools might have a subtle purpose, or they might be waiting for a new use. All these discoveries led to a huge shift in thinking. A major project called ENCODE Encyclopedia of DNA Elements really highlighted this. The ENCODE project aimed to identify all the functional elements in the human genome. It was a massive international effort. When they published their results, they made a big statement. They said that a large portion of the human genome, far more than just the protein-coding genes, shows biochemical activity. This means it's transcribed into RNA, or it binds to regulatory proteins. Now, there was some debate about what biochemical activity truly means. Does activity always equal function? Maybe not always, but it certainly suggested that a lot more of our DNA was doing something than we ever imagined. It wasn't just silent. It wasn't just sitting there idly. It was being read. It was interacting. It was doing things, even if we didn't always understand the exact purpose yet. It was like finding out that the blank pages in the book weren't blank at all. They had invisible ink, or they were textured, or they were specially bound to affect the other pages. From an evolutionary perspective, the existence of so much non-coding DNA also makes sense. It provides a huge playground for evolution. Changes in protein-coding genes are often dangerous. They can break an essential protein, and that can be fatal, but changes in non-coding regions are often less risky. They can accumulate, they can be tested, and sometimes they can lead to something useful. They can create new regulatory elements. They can fine-tune gene expression. This can lead to new traits or better adaptations. Think about how different species are. Humans and chimpanzees, for example, share almost 99% of their protein-coding genes. Their proteins are very similar. Yet, we are very different animals. A lot of those differences might come down to how those similar genes are regulated. When are they turned on? How strongly? In which cells? Non-coding DNA, especially enhancers and LNCRNAs, likely play a huge role in these differences. They are the conductors that make the same instruments play very different songs. The sheer bulk of non-coding DNA also influences the three-dimensional structure of our chromosomes. How DNA is folded inside the nucleus is not random. It's highly organized, and this organization affects which genes are accessible and which are not. Non-coding DNA can help determine this genomic architecture. It's like the scaffolding and walls of a building. They don't make up the furniture or the people inside, but they determine the layout. They determine how the building functions. So the term junk DNA was a temporary label. It reflected our ignorance. It reflected the limitations of early science. But as our tools improved, as our understanding deepened, we saw more and more purpose in those once ignored regions. We realized that life's instructions are more complex than just a list of ingredients. They also include a very detailed recipe book, with instructions on when to mix, how long to cook, and what temperature to use. It's like thinking a computer program is only about the lines of code that make the program actually do something. But it's also about the comments, the formatting, the spaces, the way the code is organized. All that non-coding stuff makes the program readable. It makes it maintainable. It makes it work efficiently. Without it, the functional code would be a mess. It would be impossible to understand. It would be impossible to fix. We are still in the early stages of understanding much of this non-coding DNA. It's like looking at a vast, unexplored continent. We've mapped some of the rivers. We've found some big mountains, but there are still huge areas we haven't explored. There are still many mysteries. What exactly do all those thousands of LNCRNAs do? How do all these regulatory elements interact in such a precise way? How do these elements contribute to individual differences? or to disease susceptibility. This ongoing research is vital. It's leading to new ways of thinking about biology. It's leading to new ways of understanding diseases. Many diseases, like cancer, heart disease, and neurological disorders, are not just caused by faulty protein coding genes. They are often caused by problems in gene regulation, by things going wrong in the non-coding regions. So, understanding the real purpose of this so-called junk, DNA, is crucial for developing new treatments, for finding new cures, for truly understanding human health. The story of junk DNA is a humbling one for science. It reminds us that what we don't understand today might be incredibly important tomorrow. It reminds us that complexity often hides in plain sight. It teaches us to be cautious with labels, and to keep exploring, to keep questioning, because the more we look, the more we find. And what we find is a genome that is far more dynamic, far more intricate, and far more purposeful than we ever could have imagined. It's not just the words in the book, it's the punctuation, the layout, the chapter headings, the blank spaces, the illustrations. All of it works together. All of it is part of the story. All of it has a purpose.